Hello and welcome to today's Saturday session for biology. And the focus for today, we're going to look at DNA, the replication and profiling, genetic engineering, and then we're also going to have an exam overview at the end, just with all the changes that are coming in with this year's exam. So just a reminder to follow us on social media. So this is where you'll find all of the updates about our revision courses coming up for Easter and all of the information about our other Saturday sessions. So again, just an overview of today. The first 40 minutes or so, we're going to have a look at DNA structure, replication and profiling. And then for the last couple of minutes, we'll have a little bit of an exam overview and tips so that you can do as well as possible in your final exam. So we're going to start off with the basics and have a look at the structure of DNA. So DNA stands for deoxyribonucleic acid. Okay. It always takes the form of a double helix, which we can see over here in the picture. So if you picture a twisted ladder, that's what DNA is going to look like. So it will have two backbones here. So one of them is pictured in blue, one of them is pictured in red, and then all of our base pairs, which we'll come to in a couple of minutes, will be here in the center. Now the basic unit of a DNA, because DNA is just not made up of one singular thing, it's made up of loads of tiny little things, which are basic units called nucleotides. And all of these have the exact same generic structure. So we have a sugar in the center. This is the deoxyribose. Nucleotides will always have a phosphate group at the side, and they're going to have a nitrogenous base. And that base can be one of four things. It can be adenine, guanine, cytosine, or thymine. And we'll come to those in a couple of minutes. But just to remember, they all have this sugar, deoxyribose, they all have a phosphate. It's just the nitrogenous base that's going to change from nucleotide to nucleotide. Now, as I'd mentioned, there's four base pairs which a nucleotide can have. So they're adenine, guanine, thymine, and cytosine. Now, these are usually broke down into two separate groups. So adenine and guanine are known as the purines, whereas thymine and cytosine are known as the pyrimidines. And if we have a look at the diagram over here, this is just a subsection of a double helix. And you can see here, if we look at where the bases join, T and A are what we call complementary bases. So they're the ones that are going to join together. And you can see here that the end of T compared to the end of A, they actually fit in perfectly. And it's the same for G and C. So A is always going to join with T, G is always going to join with C. And if you're looking for a way to remember that, if you just picture this down here, so it's a picture of the Giant's Causeway, and just remember at the Giant's Causeway. So again, adenine joining to thymine, guanine joining to cytosine. Now we've mentioned that DNA always takes the form of a double helix. So the base pairs are going to begin to join. And as they join the whole way up, it's eventually going to pull the two strands of DNA into the double helix shape. So we've already mentioned that adenine is always going to join with thymine and guanine with cytosine. But in here at the center, to make sure that they're going to be held together, they're joined together with hydrogen bonds. So important to remember that it's hydrogen bonds that are doing that. So if you just remember the H at the start of helix and the H at the start of hydrogen. Now, there was a couple of scientists who did a lot of work on the investigation of the structure of DNA, and that was Rosalind Franklin, Crick, and Watson. So they all worked on the idea of the double helix structure, where the outside strand, so the bit that's coloured in grey, is the sugar and the phosphate, and the inside is the base pair. So Rosalind Franklin was one of the people who did a lot of the initial work, but Watson and Crick subsequently got a lot of the recognition for it. So important to remember there that Rosalind Franklin had a massively important role in the discovery of the structure of DNA. Now, why is this important for us, and why does the order that the base pairs join in matter? Well, along the strands of DNA, the base pairs are going to appear. So we can see here they're all towards the center. Now, this DNA is going to code for protein using 3 base pairs at a time. And each sequence that we're going to have is known as either a triplet or a codon. So each triplet, or each 3 base pairs, is going to encode for 1 amino acid. And these amino acids are the building blocks of proteins. And if we have sequences of these amino acids, so chains of these amino acids joined together, that's when we're going to form our proteins. So if we were to have a look over at this table down here, we know that we're dealing with triplets. So we need 1, 2, 3 base pairs. So let's say, for example, our base pairs were A, T, C. So we're going to look for A in our first base pair. So there's A. T is the next one. So these are all the ones that begin with AT. And then we're going to look for the one that ends in C. So ATC is going to encode for this particular protein. Now, some of them actually aren't going to encode for any proteins. And if this does happen, this is what we refer to as junk DNA or non-coding DNA. So all along, if we have a look at the table over here, we'll see that we have some stop codons, and again, we'll have some start codons. So if we were to have TAA or TAG or ATA, that means that we're going to actually stop coding for protein, and we're not going to start again until we get to a start codon. Now, very similar to that, we have RNA, but there is a couple of differences between DNA and RNA. So RNA stands for ribonucleic acid, so it's a different sugar. Instead of having deoxyribose, we have ribose. RNA also has 3 of the same base pairs as DNA. So we have A, we have G, and we have C. So we've adenine, we've guanine, and we've cytosine. We don't have thymine, however. Thymine is going to be replaced by uracil. Again, a little difference between DNA and RNA is rather than being a double helix, RNA is just single-stranded. And it's always going to be complementary to the base pairs in DNA. So, for example, if we have a DNA sequence that was AGA, CTA, A, as we know in DNA, would join with T, A for at the Giant's Causeway, so AT, but we don't have thymine in RNA, we have uracil, so we're actually going to join with U. We will have G, that's going to go with C again, A back to U, C with G, T is going to join with A because we do have adenine in this case in RNA, and A again, we don't have thymine, but we do have uracil. So it's always going to be complementary to the DNA, but just remember there's no thymine, it's replaced by uracil. And we can see that in the diagram over here again. So here's we have our DNA strand. The complementary strand of RNA has to be matched up with the DNA. So C with G, U is going to go with A, U with A, and so on. Now here's just an exam question that has come up on DNA before. So what do the letters DNA stand for? Well, we know now that's deoxyribonucleic acid. Part B asks us to identify molecule G. So if we go up here and look for molecule G, well, we can see here it's been bonded to C. C is cytosine, G has to be guanine. Part C now says, the structure labeled J is a subunit of DNA. Identify the structure labeled J. So we can see here it's composed of 3 different parts. There's our sugar, there's our phosphate group, and there's our base pair, which is cytosine in this case. And the 3 of those together form the basic units of DNA, and that basic unit is a nucleotide. Part D, name the part labelled K. So if we go over here, we can see here that we've 2 base pairs being joined together. We've adenine and thymine, so we can say base pairs, or even better, we can say complementary base pairs. Part E, name the bonding that occurs at L. So again, we're dealing with a double helix, H for helix, H for hydrogen bond. Part F, state one structural difference between DNA and RNA other than the number of strands. So we can say here that thymine in DNA is replaced by uracil in RNA. And finally, give one non-nuclear location of DNA in cells. For that, we could say the mitochondria. So another exam question here. Part 1 says DNA is made of units called nucleotides. Draw a label diagram of a nucleotide to show its 3 constituent parts. So really quickly, we know that it's going to have its pentose sugar, so 5-sided sugar in here, deoxyribose. It's going to have a phosphate group. And just make sure we're really explicit with our labels. This is our sugar. And then finally, we're going to have our base pair. So you can just draw any generic structure for this. That's our base pair. Okay, so part 2. Which of the labeled parts in your diagram may vary from nucleotide to nucleotide? Well, we know that's the base pairs over here because it can be adenine, guanine, cytosine, or thymine. So that's our base pairs. Part 3. Genetic code is contained within the DNA of chromosomes. Briefly describe the nature of this code. Well, we can say that base pairs will appear as codons or triplets. These will encode 4 amino acids. And then these amino acids will form proteins. So when we have chains of them, we're going to form our proteins. And then they will be expressed in humans. So I'll go up here to the top for part 4. What is meant by non-coding DNA? Well, that's just DNA that doesn't encode for proteins. Also known as junk DNA. Part 5, give one structural difference between DNA and RNA. So previously we had mentioned the one about how RNA has uracil rather than thymine, but here we can say DNA is double-stranded. But again, not enough just to say what the story is with DNA. We have to mention RNA there as well, because we don't know whether it's single-stranded, triple-stranded, what is it? We have to say it's a single strand. So just make sure you give both sides of the story there. And part 6, name a cell organelle apart from the nucleus in which DNA is found. And again, you can see it's a very common exam question there. We could say the mitochondria. Now that was the generic structure of DNA. We're now going to have a look at the replication of DNA. So if you think back to interphase in cell division, and in particular mitosis, before the cell can actually divide, it must replicate the DNA within so that it can be passed off to the daughter cells. So we can see in the diagram over here at the bottom, interphase actually accounts for a massive proportion of the cell cycle. So we can see here replication is going to occur and then the synthesis of new organelles, but we're going to focus in on the replication. It occurs in the nucleus because this is where the vast majority of genetic material is found. Now we're going to have a look at the replication of DNA, and there's 4 steps which we're going to see, and I'm going to explain them both in text but also in the diagram down here in the bottom. Now if we have a look at the first piece of the diagram, we have our original DNA strand, it's a double helix, we have our base pairs in here in the centre, joined together by hydrogen bonds. Now remember, the goal is we want to replicate the DNA, so we want to produce new DNA. So how we're going to do this is the original DNA is going to be pried apart. So it's going to unwind and enzymes are going to break the hydrogen bonds that exist here in the centre. What are we left with? Well, we're left with the 2 original strands, one here and one here. Now existing base pairs are going to enter into the nucleus and they're going to start to pair up with the exposed base pairs. So one up here and one set down here. And what we're left with is we'll actually be left with 2 identical DNA molecules. So we're left with double what we originally had. So the DNA can actually replicate itself quite quickly, and this means that when the cell does divide, it can be passed off to its daughter cells. So what happened? Well, the double helix unwinds, enzymes will break the bonds between the base pairs, existing base pairs will come in and join up with the original strands, and then they will wind up to form a new double helix, and we'll actually be left with double what we originally had. Okay, so here's an exam question based off of that. Again, it'll be very similar to the ones that we seen a couple of minutes ago. So name the sugar present in DNA. So we know that's deoxyribose. Part 2. Other than the sugar, give 2 structural differences between DNA and RNA. So we're not allowed to mention the sugar in this case. So we can say DNA is double-stranded, RNA is single-stranded. So just make sure I mention what's in both. And DNA has the base pair thymine. And this is replaced by uracil in RNA. Okay, down to the next part. Name the biologically active entities, each of which contains only one type of nucleic acid. So in this case, this would be a virus. And part 2, name 2 locations in eukaryotic cells where RNA but no DNA is found. So in this case, we'd be talking about the cytoplasm, and also we could say the ribosomes. Now, with all the cell division, all the replication that's occurring within our cells, it's not that uncommon for a genetic disorder to occur. So a genetic disorder is a genetic problem caused by one or more abnormalities formed in the DNA. So it can happen if the replication of DNA does not go as planned. And it can be quite simple. So if we have a look at the diagram over here, this was our original sequence. Down here, this is one type of mutation which can occur. It's called a point mutation because a mutation will happen at a very particular point. So we can see here, if we relate the two diagrams, that's that particular point where it's occurred. Now, genetic screening can test the DNA for the presence or absence of particular genes. So it can actually test whether a genetic disorder has occurred. So it can be used in adults to test whether they will pass disorders to their children. So in relation to that then, we also have DNA profiling. So DNA profiling is a process where a specific DNA pattern, called a profile, is obtained from a person or sample of bodily tissue. So we can see here, these are all different profiles. Okay, so in a particular crime scene, this was the profile that was gathered. We would then match this to particular profiles that were got from suspects, and we're looking for matches along the profile. So if they have these bands in roughly the same location in each one, then we can say that it's coming from the same person. So it's generally used in forensics and can also be used medically in paternity tests. So there's 4 steps within DNA profiling. The first one, well, obviously we have to get our DNA. So the DNA is going to be extracted. We can't obviously analyze all of the DNA. We're going to only analyze a sample of it. So the DNA is going to be cut. Fragments within the DNA are going to be separated because that's what's going to actually produce the profile. And then the final thing we're going to do is we'll compare the patterns if it's at a crime scene with the suspects and maybe off the body at the crime scene, or in paternity tests off the father and then off the potential child. So again, what happens within each one of these steps? Well, in the extraction stage, so the DNA is extracted, we're going to break down the cells. So cells are broken down to release their DNA. Following this, with all the DNA being exposed, we're going to cut the DNA. So again, enzymes are going to do this for us. So they're going to cut the DNA into small pieces. And we can see that happening over here. Now, the next stage is probably the most complicated stage. And this is where the fragments will be separated based on their size. And that's what's going to give all of the profiles down here. So this is done based on their size. The process, which is generally used, is called gel electrophoresis. So the DNA is placed into a gel and an electric current is passed through. So this is going to pull the DNA from one end right down to the other end. Because the smaller fragments are able to move that little bit faster and further, they will appear further along the profile. So the bigger fragments will remain towards the start. Now, once we have all our profiles down here, we're then able to compare the patterns. So there's a very, very slim chance that 2 people will have identical profiles. So if the patterns from 2 separate samples match, then we can definitively say they're from the same person. So again, at a crime scene, we can say that it's either the suspect compared to the body, or if it's a paternity test, it's the father and the potential child. So here's an exam question based off DNA profiling. So the series of images below represents the 4 main stages of DNA profiling. Part 1, distinguish between DNA profiling and genetic screening. So for that, well, we've seen DNA profiling. So profiling generates a pattern of bands, whereas screening tests for the presence or absence of genes. Now, part 2, explain each of the stages 1 to 4 in DNA profiling as shown. So, stage 1, we have the isolation. So, DNA is released from cells. Part 2, we have cutting. So DNA is cut using enzymes. Part 3, we have separation. So fragments are separated based on size. And if you wanted to mention there as well, gel electrophoresis. That was the process. And number 4, I'll fit it in here at the bottom, we have comparing. So patterns are compared. Okay, so that's the 4 stages. Now I'll actually go back up to the top here just for part 3 and part 4. So give any applications, or any 2 applications of DNA profiling. So we could say paternity tests, and we could also say forensic scenes. And finally, identical twins have the same DNA profile. Explain why this is so. So for that, because they're coming from the same zygote, we can say they have the same genes. Now, the final part of DNA we're going to look at today is genetic engineering. So genetic engineering is the artificial alteration of genes. So the altered DNA, or the DNA that we've changed, is sometimes known as the recombinant DNA. So it's just important to remember that as well. And it's always going to involve 2 cells. So the process is going to need our human cell, and then usually the second cell that we're going to work with will be a bacterial cell. Now there's 5 main steps to genetic engineering. We have our isolation, which is very similar to DNA profiling. Again, cutting is very similar. It's the second stage, but then we have different stages. We have transformation, we have the introduction of base sequences, and then we have our expression stage as well. And we can see in the diagram over here the 2 cells that we're going to be involved. We have our human cell, and we also have our bacterial cell, and both are vital to the process of genetic engineering. So a little bit more detail about the steps involved within genetic engineering. So the first one is isolation. So we need to find our target gene. What's the gene we want to isolate? So the target gene on the DNA is identified, located, and it's removed from the cell. And we can see that down here in the diagram. So we've identified our target gene, it's after being removed. At the same time, the human DNA and the plasmid DNA, so the plasmid DNA is coming from the bacteria, and we can see that down here, it's after being cut, so we have a missing segment. They're both cut using restriction enzymes. Transformation then is going to involve the 2 DNA segments being combined together. So we can see here, this was the section that was cut from the human DNA. It's been placed into the bacterial DNA, and we'll be left with a bit here that has both. Now, step 4 is the introduction of the base sequences. So the host DNA is now going to begin to change. Okay, obviously this is going to happen because we're combining 2 different types of DNA. And then finally what's going to happen is the organism with the altered genes is now going to express the required proteins. So we'll just recap all that in the diagram. We've identified our required gene here in the human. That's going to be cut. At the same time, the bacterial DNA is going to be cut. And at stage 3, the transformation, they're going to be combined together. So we can see here the majority of it is still the bacterial DNA, but a segment of it is this section of the human DNA which we've put into it. Now all of that is put back into the bacterial cell, and everything within that now is going to begin to change. So the DNA is going to begin to change, and it's now going to begin to express the required proteins. So we'll see in a couple of minutes why this might actually be used. It's particularly important for diabetes patients and also within food production. But this bacterial cell is now going to express the proteins that we want. Okay, so where is this actually used? Well, one of the ways that this can be used is to make insulin for diabetes patients. And we can see that the exact same stages are going to occur over here in the diagram. So we have an insulin producing cell within our bodies, and we have our bacterial cell with the plasmid. So the DNA is going to be removed, and we're going to use restriction enzymes, and we're going to cut out the gene that produces insulin or responsible for insulin production. At the same time, the plasmid is going to be removed from the bacterial cell. It's going to be cut as well, and we're going to have transformation occur down here. So the plasmid is going to be combined with the insulin gene, and we're now going to have a new piece of DNA down here. What's going to happen? Well, this is going to be placed back inside the bacterial cell, and that's now going to be transformed, and it's going to start to produce insulin. This insulin can then be taken out and used to treat diabetes patients. So if we just have a look at the text that's wrote over here, the gene for insulin production is placed into our bacterium, which then produces insulin, which can be removed. Another way that this can be done is crops can actually be edited to become resistant to weed killer. This is particularly important for crop production. So if you can imagine, we have vast areas full of crops. If we were trying to spray all the weeds within those fields, very, very easily we could actually kill the crops as well as the weeds. But if we were to genetic engineer those crops, well, it means that we can spray all around the crops and not have the fear of killing the crop. It's only the weeds that would actually be killed. Now, all of that obviously gives rise to particular ethical concerns, and some of these are animal welfare, the idea of designer babies, which I'll come back to in a second, and then unanticipated welfare concerns. So animal welfare comes back to the idea that up until years ago, animals would have been hugely involved in the testing involved in genetic engineering. A lot of the time now it is bacterial cells, but in the past animals would have been tested on. Designer babies is a really interesting conversation that happens around genetic engineering. So essentially, a designer baby is a baby whose genetic makeup has been selected or engineered just to make sure that certain traits are present. So potentially you'd be able to select for eye color, hair color, height. You might be able to eliminate or lower certain health risks, potentially look at athletic ability, musical ability, the intelligence, maybe even the sex of the baby. So it does give rise to the idea that, is this ethical? Should we be allowed, or should we be able to choose certain traits that offspring are going to have? So it's a very interesting debate, and there's lots of information readily available online if you wanted to look a little bit more into that. And then obviously we have unanticipated welfare concerns as well. So a lot of the tests are done short term. We don't know whether this is going to have any lasting effects. If we have genetic engineered crops, they're quite a new phenomenon. So mightn't actually see any effects on our bodies as of now, but is that going to be the case in 20, 30, 50 years time? We don't know. So there is welfare concerns that maybe we haven't anticipated just yet. So a couple of questions based on genetic engineering as well. So what is meant by genetic engineering? So this is the artificial alteration of genes. Now the second one, genetic engineering involves the following steps, but not necessarily in the order given. Rewrite these steps, placing them in the correct order. So just before I get into this as well, really, really important on a question like this. On the day of the exam, if you're really unsure, make sure we're guessing this question. Really, really important that we don't leave it blank, because we're actually given the answers that we have to put in. So definitely make sure we're guessing if we're a little bit unsure. But the steps involved, we have isolation. So the target gene is going to be isolated. We're then going to cut. We're going to have transformation. So it's going to be combined with the bacterial DNA a lot of the time. And then it's going to express our desired protein. So briefly, what is meant by isolation and expression. So isolation is identifying and removing the gene, or target gene, I should say. And expression is the production of the desired protein or product. So in some cases, it could be insulin. Okay, so the production of the desired protein or product. So again, in genetic engineering, DNA molecules called cloning vectors are used during transformation. Name the rings of DNA contained in bacteria that may be used as cloning vectors. So this is our plasmid, so plasmid DNA. Give one application of genetic engineering in any two of an animal, plant, and microorganism. So we've seen a couple of these already. An animal example would actually be to produce lactose-free milk. Plants would be resistant to herbicides or weed killers. And then microorganisms, well, we know bacteria can be used for the production of insulin. Okay, so that was genetic engineering. Now, up until now, we've looked at DNA, we've looked at the profiling, we've looked at the structure, we've looked at genetic engineering. For the last part of the session, I'm just going to focus in on the exam for 2022, the layout of what the exam is going to look like, and a couple of tips for you going into your exam to make sure that you can do as well as possible. Now, what is the layout of the exam going to be for this year? Again, as always, we've our three sections. We've section A, which is the short answer questions. We've section B, which is the experiments. And we've section C, which is the long answer questions. Now, it's very different from previous years, very similar to 2021. So section A now has 7 questions rather than 6. You're only required to answer 4, and each question is going to carry 20 marks. The experiment questions, as always, there's going to be 3 questions. You're only required to answer 1 rather than the usual 2, and it still carries 30 marks. And then the long answer questions, there's 7 questions here, so increase in choice here. We're required to answer 3 of those questions rather than the usual 4, and each question carries 60 marks as always. Now, the last 2 questions on a typical exam paper prior to the pandemic would have had part A, B, and C, and if you chose one of those 2 questions, you only had to answer 2 parts. That's actually been increased. So you still only have to answer 2 parts if you choose one of the last 2 questions, but there's now part A, B, C, and D. So there's even more choice within the paper. So all in all, rather than the usual 400 marks, the paper is now worth 290 marks. And because of that, the percentages that each section has worth towards the final grade is different. So the short answer questions are now worth 28%. The experiment questions are gone down slightly, it's 10%. And the long questions are gone up to 62%. So these are massively important for our overall grade. Now, I'm just briefly going to mention the structure of the exam, and you'll see why in a couple of minutes. But just visually, I want you to look at Unit 1, Unit 2, and Unit 3 and see how vastly different in the amount of material they all are. Unit 1 is composed of the scientific method, food, and ecology. Unit 2 is things like cell structure, cell diversity, we photosynthesis, respiration, so big topics there. DNA and RNA, we mentioned cell division there as well. Genetic engineering, they're all Unit 2. And then Unit 3 is everything to do with the plants and animals. So, you know, you've all your systems down here. You've human defense, human reproduction, plant reproduction, vegetative propagation. Don't worry too much about the content of Unit 3 as of now. What I want you to focus on is how much less material there is in Unit 1 compared to Unit 3. And that will make sense why that's important in the next minute. So if we were to look at the course structure, so Unit 1, Unit 2, Unit 3, and then compare that to the exam layout, Unit 1 can actually comprise a huge proportion of the percent of the paper. So prior to the pandemic, if we think of the exam paper, what it was like back in 2019, 2018 and so on, we would have had 6 short questions. 2 of those short questions were from Unit 1, scientific method, food, ecology. That's it. 2 of those questions were from Unit 2, 2 of those questions were from Unit 3. The experiment questions varied, but the long questions we would have had 1 from Unit 1, again, scientific method, food, ecology. It was usually an ecology question. 2 from Unit 2 and 3 from Unit 3. Now, if that holds, which it will, how is our marks going to be allocated? Well, remember, you have to answer 4 short questions. Potentially you could answer 2 of your short questions on scientific method, food, ecology. You could answer 1 of your long questions on the exact same material. So what I've said here is the importance of the table above is the fact that Unit 1 comprises of 3 topics. It's the shortest of the 3 by far, but it can account for 34% of the exam. So given the fact that the 2 short questions and 1 long question will be asked about it, my recommendation will be that food, ecology, and the scientific method, you know those inside out and upside down. They're hugely important topics. Now again, there's 22 experiments in the course, so if you combine those 3 topics with the experiments, that's 44% of your exam. Obviously then we'd be looking at Unit 2 and Unit 3 because they do form a massive part of the exam, but for 3 topics in the experiments, you're talking 44% of the exam. That's an awful lot of marks for that little material. Now, a couple of tips for each of the sections. Section A is the short questions. Generally, we're looking here for about 30 minutes. You only have to answer 4 out of the 7 questions. If you can, I would answer them all, because you are going to be marked on your best questions. And you're going to be, so I would definitely recommend to answer them all if you have time. For these, it's important to know your definitions and just get straight to the point. There's no point waffling here. You don't have the space, you don't have the time. You need to get straight to the point. And this is where exam questions are going to be your best friend. So my advice for this will be to read through all the questions and put a question mark or an X beside the questions you're unsure of, and then come back to them at the end of the exam if you have time. And if you're in doubt, guess. The only thing that's guaranteed is if you leave an answer blank, you're guaranteed 0 marks. If you put an answer in, might only be 1 word. Sometimes 1 word's enough, but if you put something in, you might get some marks. If it's blank, guaranteed no marks. And then section A, practice, practice, practice. So the questions here are so repetitive. So practice them again and again and again in the exam questions. Use your marking schemes, correct them yourself, do it again and again and again, because you will see there's an awful lot of overlap between the questions. You would have even seen it today in any exam questions that we did. Some of them were very, very similar, just worded slightly differently. The experiment questions, there's 3 questions here as always. You only have to do 1 of them though, and these questions are worth 10% of your final grade. So whatever one is your best one, that'll be marked out of 10%. You should have time again though to do them all, so I would do so if you can, and your best question will be awarded. So there's 22 experiments in total. I would recommend knowing them all in detail, but if you are struggling with the content, the new changes for the paper will actually help an awful lot with that, and I'll come back to that now in a second. And just from looking at the past experiment questions, scientific method, preparing the slides, so the animal and plant cell, the isolation of DNA experiment, the germination experiments, and all the enzyme experiments, they're the ones that seem to come up more often than others. Now, I don't actually have the 2021 exam within this, but you can see pretty much everything that I've just mentioned there holds true by looking at this. So scientific method there is a very common exam question. So too is the plant and animal cell questions. Down here we have our food test. So again, one of the topics in Unit 1 popping up. Isolation of DNA experiment is there. And we also have here the germination experiment. So we can see here they're the ones that often come up more often than others. But in the last couple of years, what started to happen is one of the experiment questions will actually incorporate maybe 4 or 5 different experiments. So you might have a part A, B, C, D, E. Each part will relate to a different experiment. But again, I definitely would be trying to answer all experiment questions if you can. Now, thankfully, if you're really struggling with content, we actually know which experiments are going to be asked in each particular question this year. So the experiment questions are going to be questions 8, 9, and 10, because short questions are the first 7. And question 8, the experiments that you can be asked in question 8 are the food tests, the ecology, so habitat studies, qualitative and quantitative studies, the preparing and examining animal or plant cells using the light microscope, and also the isolation of DNA from plant tissue. Those experiments are question 8. Question 9 is to prepare and show the production of alcohol by yeast, investigate the influence of light intensity or carbon dioxide in the rate of photosynthesis, enzyme immobilizations, the effect of pH or temperature and enzyme activity, the heat denaturation on enzyme activity, and activity to demonstrate osmosis. They are all question 9. And question 10, we have the growth of leaf yeast using agar plates, the transverse section of a dicot stem, the heart dissection, the effect of exercise on the breathing rate or pulse rate, the effect of the IAA growth regulator on plant tissue, using starch agar or skim milk plates to show the digestive activity of or during germination, and investigate the effect of water, oxygen, and temperature on germination. So if you're really, really struggling with the experiments, you know which ones are going to be asked in each question. You can make the decision which one you want to focus on. Okay, we know which ones are in question 8. We know which ones are in 9. We know which ones are in 10. So that's where the questions are coming from for 2022. And now finally, just for section C, the long questions, will be between 100 and 2 hours for this one, 100 minutes to 120 minutes. Really important you read through each question to help you decide which ones to answer. What might appear as your best question to start off with, if you start going down through the parts of the question, you might soon realize, do you know what, this isn't the question for me. There might be a better one further on. So you do only have to answer 3 questions. If you've time, again, answer an extra one. You still have your full 3 hours for the paper, so you should have lots of time. Just make sure you're including enough points for full marks as well. So everything's obviously marked in 3s. So you should be able to give somewhat of a guess of what a question is going to be worth. But again, if it's a 6-mark question, you're going to need 2 points. So just make sure we're giving enough information. And very important for questions that ask you to compare and contrast or give differences between, it's really important that if you're given a difference, you mention what object 1 has and what object 2 has. It's not just enough. So remember earlier on when we're talking about the DNA, it said to give a difference between DNA and RNA. It's not just enough to say DNA is double-stranded. You have to say RNA is single-stranded. So give both sides of the story there. So again, what's the big questions here? Genetics, photosynthesis, and respiration are the big ones. I'm going to be focusing in on these for the ExamRevision course for anyone who's interested in doing that. And there is a genetics Saturday session already up on the YouTube if you want to have a look back at that. Ecology as well is a very common topic, and I'll include that in the ExamRevision course. And for here, again, it's practice, practice, practice. At this point in 6th year, exam papers and marking schemes are your best friend. Use these day in, day out in the lead up to the exam. And just to finish off, some general tips for your exam. Just relax. This is the big thing for students. You have to remember these exam questions have to be based on something that you've covered in class. You know, you're not allowed to be asked something that you've never covered before. So it's always going to relate to something that you've seen before. It's just going to be phrased slightly differently. Really important to read the question carefully. Is the question asking you to state or list, or is the question asking you to explain? Okay, because the difference in the question word can dictate how much you're expected to write. So just be careful of that. If you can, use a diagram. As the saying goes, picture paints 1,000 words. And sometimes this can really help with your explanation and answers as well. So don't be afraid to throw in a diagram. And if you are asked to draw a label diagram, do not forget your labels. Marks are given for the labels. Make sure the arrow is pointing exactly to what you're trying to label. Don't forget them. And for timing, if you're stuck, just move on. So the time that you're spending agonizing over that one question that you can't do is taking precious time away from questions further on that you probably do know. So just move on, come back to it at the end. And as I've said already, make sure at this point exam papers are your main focus. They're your best friend right now. So practice them over and over and over again and use the marking schemes to grade them. And just to leave you with this, just to give you a little bit of information about our revision courses. So kicked off today with Leaving Cert Spanish, and over the course of the next 2 weeks or so, you can see there's some there for history, chemistry, maths, physics and biology. So biology is going to take place on Friday the 22nd of April. It's available to book now. So if you go onto the website, there's an opportunity to book it, and you'll also see the details of the different parts of the course that will be covered on the day. So it'll be very similar to the one that we did today. Lots more content covered, lots of exam questions to practice, and even more tips and so on for your exam. So if you are interested, just make sure you get onto the ExamRevision website, examrevision.ie, and make sure and book your place now. So I hope you have a great Easter holidays. Best of luck with your revision over the coming weeks and the coming months. Just remember to keep all the tips in mind, keep practicing your exam papers, and you'll be fine. So thanks a million for joining today, guys, and best of luck.